The Structure of Life through Biopython. Python is one of the most widely used programming languages in the world today. Why do so many people use Python? One of the main reasons is its simple and intuitive syntax. Computers cannot understand human language directly. That is why we need to translate our instructions into a language that computers can understand. This is what a programming language does. The language that computers understand directly is called machine language, but it is extremely difficult for humans to understand. Python makes programming much easier. For example, if we want to tell a computer to add 2 and 3, we can simply type print 2 plus 3 in Python. Even complex instructions can be expressed in relatively simple statements. Python has another major advantage. It allows programmers to use programs and functions that other people have already created. There is a saying among programmers that we should not reinvent the wheel. If someone has already built a good wheel, there is no need to build one from scratch. The same is true in programming. By using functions that others have already created, programmers can build new programs much more quickly and efficiently. A collection of reusable functions and tools like these is called a library. So, how is Python used in biology? Biologists also use computers to analyze enormous amounts of data. One important tool for this is Biopython. Biopython is a collection of Python libraries designed to analyze biological data. It is especially useful for researchers in bioinformatics. So, what do bioinformatics researchers do? They use computers to analyze biological data such as DNA, RNA, and proteins. The amount of biological data is enormous. Imagine having to examine hundreds of thousands of DNA sequences one by one. It would take far too long. Biopython helps computers handle these repetitive tasks instead. One of the things we can do with Biopython is parsing. Parsing means extracting the information we need from a large amount of data. In bioinformatics, two commonly used file formats are FASTA and FASTQ. These files contain DNA or protein sequences. FASTA usually contains information such as a sequence name and its sequence. FASTQ also includes information about the quality of each sequence. So, how can we find only the information we need among thousands or even millions of sequences? With Biopython, a computer can quickly find the information we are looking for. The second thing we can do with Biopython is analyze DNA and protein sequences. DNA is made up of 4 types of bases: A, T, G, and C. From a computer's perspective, these are simply sequences of characters. Comparing these characters and looking for specific patterns are exactly the kinds of tasks computers are good at. With Biopython, we can treat DNA, RNA, and protein sequences as strings. We can then compare and analyze them efficiently. The third thing we can do with Biopython is automatically access biological information available online. Today, the internet contains enormous amounts of biological data. In the past, researchers had to visit websites one by one to find the information they needed. But what if a program could do that for them? A computer can automatically retrieve and organize information from online databases. Tasks that once had to be done manually can now be handled by computers. The more data there is, the more valuable this kind of automation becomes. The fourth thing we can do with Biopython is work with bioinformatics tools. One of the best-known tools is BLAST. BLAST compares DNA or protein sequences with massive biological databases. It then finds sequences that are similar to the sequence we provide. For example, imagine that we discover a new DNA sequence in an organism. We might wonder whether a similar gene exists in other organisms. BLAST can help us quickly find clues that answer this question. Researchers can then use this information to predict the function of a gene or study how organisms have evolved. The importance of Biopython also became clear during the COVID-19 pandemic. Researchers around the world rapidly analyzed viral genome data, and they shared their findings with one another. This made it possible to track genetic changes in the virus. How long would it have taken if researchers had needed to compare the genomes of thousands of viruses by hand? Computers can process and compare enormous amounts of data in a very short time. But that does not mean computers understand life for us. Computers process data quickly. Humans interpret the results. Biopython helps bridge the gap between the two. It is like a librarian who helps us find the books we need in a huge library. Once computers find and organize the data, researchers can use it to ask new questions. The amount of biological data will continue to grow in the future. So how will we make sense of all that data? This is where mathematics and computer science become essential. Biopython helps researchers organize and compare complex biological data. They can then use that data to explore deeper questions about the structure of life, the causes of diseases, and the evolution of living organisms. What new tools will help us unlock the secrets of life? The answer is not found only under a microscope. The key to understanding life may also lie in the new tools created through mathematics and computer science.